Welcome, everyone. We'll be starting 5 minutes past the hour, just to give people some time to join. Okay, hello and welcome everyone to session 2 of Epigenomic Profiling, a deep dive into chromatin mapping technologies. For those who missed session 1, you can find it available online, and I'll be sharing the link in the Q&A box along with the CEEHRC impact survey. And there's also a nice QR code that you can scan for session 1. You are also invited to submit questions throughout the session in the same Q&A box for us to answer. And now, without further ado, allow me to introduce Amin Noorani, who will be taking us on our second dive into chromatin mapping technologies. Amin is a PhD student in the Department of Cell and Systems Biology at the University of Toronto. His research focuses on epigenomics, regulatory networks, and single-cell genomics and 3D genome organization. Prior to his PhD, he gained experience as a bioinformatician at Epigenome Lab at Princess Margaret Cancer Centre in Toronto. Amin, the floor is yours.
Awesome. Thanks, Waji, for the nice introduction. So let's start our second part of the workshop. As some of you know, Waji, as some of you attended the first part, Waji explained about the technical part in the wet lab session. And today I'm going to talk about like how the analysis, how the data analysis works, and how once you perform your experiment, you get the data. So how are you going to analyze it? So let's start. As a brief introduction, there are two main families of techniques that does the similar approach, which is chromatin profiling. So there are like two main families. ChIP-seq is the traditional method, works by cross-linking proteins to DNA with formaldehyde, and you break up the chromatin into fragments and kind of like fishing out the pieces attached to your protein of interest. On the other hand, they have CUT&RUN and CUT&TAG methods, which are newer, and they give you cleaner approaches and cleaner results. So instead of breaking everything apart and fishing, you send protein like a molecular scissors directly to where your protein sits on the DNA. So it only cuts out the DNA right next to it. Cut&RUN, it uses MNase protein for the cutting part and release the fragments. But CUT&TAG uses TN5, which simultaneously cuts and tags the DNA with the sequencing adapter that eliminates a separate library prep step. So let's talk about the differences first. So we talked about the protocol, how they are different. And in overall, why bother like switching from ChIP-seq in the first place? Because the improvements are pretty dramatic. So you need far fewer cells, like thousands instead of millions. You need much less sequencing data to get reliable results. And the experiment takes 1 to 2 days instead of several days. And the background noise, which is the most important part for data analysis, is much lower. So the signal you get is much cleaner. And the resolution is also better because you can pinpoint binding sites down to exact base pair rather than the fuzzy 200 to 500 base pair window. So perhaps most importantly for those working with limited or precious samples, CUT&RUN is highly reproducible even with low cell input. So something ChIP-seq only achieves when you have abundant material. So you're done with the wet lab part. So you send your samples to the sequencing center, and now you get your results back. So you got your raw data from sequencing facility, and now you want to start your analysis. So here's the overall like full computational steps from raw to actual biology. So you start with the platform that you are. It's usually like for this type of data you're using more in R because it's more like the language is familiar, and for bioinformatics environment, R is very popular, and you can do a lot of statistical analysis easier than other programming languages, and most of the pipeline, most of the libraries are actually built in R. So you need Linux because FASTQ files, the raw FASTQ files are heavy. And also, so because they are heavy, so you need more computational resources. So most of the time you're working on a server or your cluster in your institution, or in a cloud based service like Google or Amazon. So you and then like that we get is like a command. So you need like more commands that should be installed on your computer. So we'll get there. And then you get FASTQ. So you have your FASTQ, you want to check the quality first, like before you start any type of analysis, you want to see how the data look like. So once you're sure about the quality, you go to other steps, which is alignments, and you filter out the stuff that you don't need. Spiking calibration, which is actually one of the feature of cut-and-run for normalization. And finally, we get to the peak calling, and then visualization and differential analysis. So let's talk about each of the steps. So today we're not going to going through every single metrics available, because this is not about bioinformatics course in general. So we wanna talk about how, like, as someone who worked with this type of data, what metrics were the most important ones that you should pay attention to in the beginning. And if they fail, you should, go back and think about all the factors that could involve that you get some low quality in those matrices. So usually, like, you get your FASTQ first with FastQC outputs, but if don't, you can run that FastQC as a command. It's very easy. It's just one line of code that you can copy from internet, and it's no big deal. So you run FastQC on the raw data, like a health check of the data before you start analyzing, right? So we focus on two things at this stage. So first is adapter contents. So adapters are technical sequences that added during library preparation that have nothing to do with your biology. So this is like an example of a flag adapter content. So you can see that across all the samples, each color represents a different random sample. So we're not gonna name them anything. Adapter sequences start appearing as early as position 8 to 10 base pair by the end of the read, over 75 to 90% around here, right? So it's the end here. So now they all contain adapter. So this is exactly what we need to detect before alignment. So because if these adapter sequences reach the aligner, they won't map to the genome, and you lose a huge fraction of data. And there are tools that can handle all of this. I'm just saying it's good to see and compare before and post quality check step. So this tells you how much adapter contamination exists. So if adapter content is already 0 before trimming, something is definitely off, because we want adapter at these steps, sequence of adapters. So there are like sequences that are known, so you can find them everywhere. So adapters are, they're not random sequences. And then we focus on pair-based sequence quality. So this is, for example, this is from Illumina sequencer. So they tend to lose accuracy toward the end of the rates. So once we move to the end of third 3' ends of the reads, which is normal and expects in Illumina. So the quality drops at the end of third end. So this tells you trimming is necessary and gives you a baseline to compare against post trimming. So this is the example, like what the plot looks like. So in this case, you can see the quality dropping toward the third prime ends, which is typical in Illumina sequencing, and exactly what we use trimming to correct. So this doesn't mean once you see this stuff, it's failed, except the like 0 like adapter content. It's just that you need to perform quality check after checking the FastQC results. So trim-galer is a wrapper around CUT&ADAPT. CUT&ADAPT is another tool. Maybe you've heard of it before. Similar to FastQC, they're from same team. So this trim-galer has both of the tools together. So once we know what's in our raw data, we trim them, and we use trim-galer to cutting all the unnecessary information, unnecessary sequences in our data. And you don't want to, do all of this manually. Of course, trim-galer handles that. So it removes the adapter sequences from the end of the reads and also trims low quality bases at the third, the 3' ends, as we talked about. So that's where the Illumina quality tends to drop off, right? So after trimming, it automatically runs FastQC, so you don't have to do anything, right? So it automatically runs FastQ again, so you can confirm the adapters are gone and quality has improved. So you see, like, it's just what positions we are removing, right? So the blue and the orange one on both sides, they're like adapter primers, and what's left in the middle is actual biology we care about. So in practice, we always check the pre and post trimming read counts in MultiQC, which is, another tool for quality check, but we'll talk about that later. So if you lose more than 10 to 15% of reads during trimming, that's a flag and it's worth investigating. So you cut all the unnecessary information and unnecessary sequences, all are removed, and you're ready for alignments. So you align your reads to reference genome, essentially finding where each piece of DNA came from, right? So bowtie, many of you heard of it for sure, is the go tool for at least for ChIP-seq, CUT&RUN, ATAG, or all these assays. Is the standard one because it's fast, memory efficient, and works well for short reads against large genomes like human and mouse. And when we say fast, doesn't mean like minutes, we're talking about hours. Fast in terms of in comparing to that large amount of files, that we can call it fast, but not the fast that we know in something you run and it just finishes quickly. So after alignment, so you align your reads to the reference genome and you get piled up reads over your reference genome. Okay, so you want to see what are the duplicates here, right? So I mentioned this because this is important after alignment. So we run Picard mark duplicates. So this identifies the reads that are exact copies of each other, likely from PCR amplification during your library prep. But importantly for CUT&RUN, unlike other assays, we mark the duplicates, but we don't remove them. This is very different from other assays because MNase cuts at very specific positions, right next to your protein. So two fragments with identical coordinates might, genuinely represent two independent molecules from two different cells, not like PCR artifacts, right? So removing them would erase your strongest biological signals. So Picard gives us the duplication rate as a quality metrics, but we keep the reads in the downstream analysis. So duplication rate, unlike other assays, you get high duplication rates and you think it's a red flag, but for CUT&RUN, you should be careful about that. So that's why Picard mark duplicates is here. So it doesn't remove them. So It's just that you have to check manually by yourself, but we'll get to that later. So after alignments, what do we get? Like the output file that we get. I'm sure many of you know about that, or I don't know. That's you heard of like the BAM files, right? So BAM files are the binary alignments or map files. So all the reads, as we talked in previous slides, are piled up on the positions that are aligned to the reference genome. So it's good to mention like what information they are there. It's more like the position. It's more like, you know, dense files that has like a compressed table with one row per read. And you use this file for almost all the downstream analysis. So if you want to visualize it, it looks like this, you know, like all the reads are here, and that's the reference genome for visualization. So it's good to know what's inside them. And they have like quality score and the orientation and everything. So all the information about the alignments of your samples. So you align your raw data to your reference genome. So after alignments, then you have other metrics to check. So you check three things. First, did you get enough data? So cut&run is clean so that you just need 1 million reads. Sometimes it's enough for, like, it's like far less than ChIP-seq, right? So 1 million can be enough for you. And the second is, did your read actually maps? So sequencing depth is important to for the quality check, but you should consider that you know, number of cells are a bit like far lower, and it's not like if you did ChIP-seq before, it's not like them, so you have to consider that. And second, you have to check the alignment rates. This is the metrics that you should not skip if the number is below some kind of threshold. So we want more than 80% of the reads to map successfully. And if it's lower, then something went wrong in the experiment or data quality. So it's not kind of like acceptable below 80%. And also the duplication rate that I talked about. So let's say like, okay, so we talked about what duplicates are, right? And we normally remove millions of unique, you know, like in ChIP-seq, for example, you have millions of unique fragments from high input library. So if you see like the same fragment 10 times, it's very likely those are PCR copies of one original molecule, not like 10 independent molecules that happen to be cut at the exact same position, right? So removing them is safe, at least from a statistical point of view, because the library complexity is high and the true biological duplicates are statistically rare. But in cut&run, as I mentioned, because the cells are fewer and you need fewer reads, this means the library complexity is lower, and the feature of MNAs, which is cutting the very specific positions right next to your like TF binding site, for example. So two fragments can be identical, can have like identical coordinates. So we say like ideally we want duplication rate to be lower than 20%. But from my experience, I saw a lot higher duplicates that were like actually from biology, not like the duplicates that we all know about, right? So it's okay to, you know, move forward when you say it's not like alignment rate, right? So when you say it's like up to like 80% duplicates, you should still move forward and check other metrics of your data. And if they are fine, then you probably don't need to be worried about this number. But like more than 90% is like, that's too much. So you gotta, further investigate into your data. And another feature from CUT&RUN protocol is the spiking calibration, right? So like normalization. So we want to fairly compare signal levels across samples, and we want to normalize them as other assays, right? So raw read counts aren't directly comparable because one sample might have, simply have been sequenced more deeply, right? This is where the spiking normalization comes in. So in CUT&RUN, in CUT&TAG, sorry, the TN5 enzyme is made in bacteria, E. coli bacteria, and accidentally carries tiny bits of bacterial DNA. So since you use the same amount of enzyme in every sample, you're adding the same amount of E. coli DNA to every tube for, no extra step is needed, right? But in CUT&RUN, we don't have that luxury. So there is no such like carryover. So we manually add fixed amount of foreign DNA ourselves. So same logic, just done by hand. But either way, the math is simple. So you calculate scaling factor based on how many E. coli reads you got back, right? A sample with more E. coli reads relative to signal was less efficient. So you scale it up. A sample with fewer gets scaled down. So everything ends up with like the same ruler. And you heard about like other normalization methods like RPKM or CPM. So they're like depth normalization method. So they assume the total amount of protein binding is same across samples, which is exactly what you are trying to measure. So, and therefore a circular assumption, right? But the spiking normalization uses an external reference, which is completely independent of your biology. But there's like a caveat that's worth mentioning, which is this only works when comparing sample processed with the same batch of enzyme, I guess, right? Maybe you know that already, but me as a bioinformatician, I think it's important to sometimes, take that into account because different batches carry different amount of DNA. So it should be same batch, same amount. And interestingly, this method is going to be used in other assays like ATAG, because I'm hearing from like colleagues that are using it for their normalization. Because it's more reliable, I guess, yeah. Okay, so you have you tested everything. So like you checked everything, right? You have your metrics all passed, and now you come to the complexity metric. So this tells you how many unique DNA molecule you actually capture, because we want to make sure like how many of the unique molecules of unique DNAs are present. So if you sequence a lot, but your estimated library size is tiny, like 1 to 2 million unique fragment, it means you just keep reading the same fragments repeatedly, right? So that's a sign of either a poor experiment or that you sequence far more than you needed to. So we estimate this using a tool called PSEQ, which generates the complexity curve you see on this slide. This is like an example from MultiQC. So in x-axis, you show the total reads sequence, and y-axis show the unique molecule captured. A curve that rises steeply and keeps going, like the dashed line, means your library is rich, and sequencing more would give you new information. So a curve that flattens out early means you've hit the saturation. So you're just rereading the same fragments, and additional sequencing would be wasteful. But this is like, it depends on the question that you're asking, right? Sometimes you don't care, but it's good to just care about, just see what's happening in terms of like the complexity of your library. And the fragments length distribution. So you get fragments, and you want to see the distribution of the length of those fragments, because like this is important, because DNA in the cell, as you all know, even like better than me, DNA in the cell, they're like wrap around nucleosome, right? So each about 180 base pair long. So when cut-and-run cuts near those spools, you get fragments 180 base pair, like 180, 360, or 540, and so on. So you should see those peaks clearly in the distribution, as you see here. Just like you can verify like what are the length of your fragments. For transcription factors, you also see like a faint like 10 base pair repeating pattern caused by the helical twist of the DNA, like a subtle staircase in the distribution. So both patterns are hallmark of successful and specific experiments. So if you don't see that pattern, if your distribution looks flat or randomly, you see like jumps and stuff, then it's a strong sign that, for example, your MNA is cut non-specifically, meaning the antibody likely didn't work as expected, right? Dealing with antibody stuff is difficult, as I And then peak calling, right? So peak calling is important in assays like ATAG or ChIP-seq, and now CUT&RUN. But first, I'm gonna mention about another file format for those who doesn't know, which is about the bed file. Bed files are just a table that is kind of like a text file that have 3 column minimum. Chromosome start and end. So it's just show like start and end of like the position, like the coordinate of certain region in the genome. So you get your BAM file, which shows the reads align on the reference, and you get the peak files, the ones that like it reads all these piled up reads, and by different statistical analysis finds out which one are actually significant and not random, and the ones that are high, but there might be some other stuff like happening, not just the one that you are looking for, like a binding site. Sometimes they're like some just random, right? So you want to make sure the one that you have is significant. All of this is happening in the background, right? So you run a tool and that does everything for you. So we'll get to that in the next slide about different tools. So peak calling, it just go from a pile of reads aligned to a genome location. And the idea is wherever reads pile up significantly above background, that's a peak. Yeah, it's more about like statistics rather than biology, honestly, for this app. And BED helps us to make it more simpler and don't need to deal with BAM file when we want to check where in the genome we have like a binding site, for example. So this is also like typical for all ChIP-seq, ATAC, and all these assays. So because the nature of the signal and background are fundamentally different from each other in ATAC, CUT&RUN, and all those assays, that's why we should be careful about the peak calling tool that we're using. So speaking of peak calling tools comparison, there are 4 main tools. First one is Max2. Many of you heard of that. It's a model-based analysis. It stands for model-based analysis of ChIP-seq, but this is the version 2. We have also version 3 of that. It was originally built for ChIP-seq and uses a statistical model, and it assumes that reads follow a Poisson distribution. That's what the method is about. You don't have to know what that is. So it works, but it works on ChIP-seq type of data, not. I mean, it works for CUT&RUN as well. Okay, let's rephrase that. But before that, we need to know about the background, right? So we want to know the peak that are called or not the background. So what is background? So in ChIP-seq, for example, you cross-link everything, right? So you sonicate the entire genome into fragments, and then your antibody pull down. But the pull down is never like perfect. So you always like precipitate random chromatin fragments that have nothing to do with your protein of interest. So these random fragments are like mapped all over your genome, creating a low-level signals everywhere. That's what we call background, right? So if you had like experience visualizing ChIP-seq data, you see it's like enriched all in so many random places, and then in binding sites, maybe there are like some enrichments, more like enrichments you can visualize by eye. It's because of that, because ChIP-seq has a lot of background. So Max2 was designed for this type of data, right? But on the other hand, CUT&RUN does not have this type of, the nature of the data is not like that, right? Because it's known for low background. So, which means it can overcall peaks by mistaking the naturally sparse signal for enrichment above a noisy background. Doesn't really exist. So my suggestion is always try to find out like what methods exist. So because of that, CEEHRC was developed. It stands for Sparse Enrichment Analysis for CUT&RUN. So it uses the actual data distribution. So first it looks into the data, and it sets a threshold empirically, which much better fits. And usually for CUT&RUN, it's more like popular because it was designed for CUT&RUN, and also you can input your IgG, which is the negative control, so it takes that into account as well. And also you can set a threshold, like test with different threshold, so which is ideal for some kind of data. Another one is GoPix. So GoPix uses machine learning and tends to be more conservative. So it's only call the peaks that are high confidence. So it does not call anything that is not, above a threshold. So it's good for like data like transcription factors, for example. It works perfectly, and it's getting more popular. but depends on like the data that you have. You can test each of them separately to just make sure what you are getting is like they're giving you what you want, right? Because they are not perfect. They are also some tools. So I mean, it's not nothing is perfect in the world, right? So you gotta try everything to get what you want. There's another tool called CUT&RUN. It's worth mentioning because it uses deep learning and it's very sensitive. So it catches like all the weak signals as well and potentially introduce, I mean, more false positive peaks. So the first peak calling method for CUT&RUN is like the ideal one is CCARD, and you can test with other tools like GoPix and Max2 to make sure like you can test all of them and see what peaks they all agree to, and then this is kind of like good to test everything for peak calling method, because peak calling is tricky because of the background, as I mentioned, and the statistical method they use in the under the hood, right? So you call your peaks and you have your aligned file, your alignment file, you have your peak file. If you want to make sure the peaks are actually in the right positions, this is like another feature, that another metrics that you can check how confident your peaks are. So FRIP stands for fraction of reads in peaks. Very powerful metric. Out of all your mapped reads, what fraction actually land inside the call peaks, right? So if most of your reads are just scattered background noise, your FRIP will be low. A FRIP above 3 is considered good by ENCODE standards, and above 2 is acceptable. This is like an example from like how they are mapped by multi-QC again. So multi-QC, just like fast QC, it gives you more information about post alignments, like pre and post alignment, but not like fast QC. It gives you a lot of information and all at once in a single report. So all the plots that I showed you, they are all generated from multi-QC. It's worth to mention that. But this sample, it does not have a good quality. So this is like also very common that this happens. Yeah, like FRIP is also like, it tells you like if the data is poor or, okay, no. Let me mention that there is like a caveat here. So FRIP is not just measure, it's not just a measure of data quality. It's also a sensitive to how you ran your peak color. So even the highest quality data will show a low FRIP score if your peak calling threshold was set very stringently. So simply because fewer peaks were called, right? So you have to be careful when you're judging your quality based on all these matrices. Some of them are obvious. Some of them are like from raw data. You didn't do anything. But in this step, you applied so many tools to your data. So it's important to know what happened before that data is generated. So it's not like I see a score is low, then call it a day. Of course, it's not that easy. So you go back and check your peak calling method, the threshold, and everything, and play around with all those tools to see how you can change it. Like peak calling, of course, if you set the threshold very low, like very significant, then it gives you lower, like more confidence peaks. So it's important to know that as well. And also it's important to check everything manually, not just by trusting all these results from these tools, because they are not perfect, as I mentioned. And finally, you get the replicate reproducibility. So you're happy with everything with like individual sample quality metrics. So you want to ask some, like, the point of the replicates. What are they? Do our replicates actually agree with each other? Because if two replicates from same condition look completely different, something went wrong and you can't trust your results. This can happen in, you can apply this in the beginning after the alignment with the BAM files that you generated. Yes, make sure that their replicates are actually replicates, and the methods that you use for correlation, like testing the correlation, is the Pearson and Spearman. So they tell you how similar your data through replicates are by, like, in unsupervised way. This is critical because you want to make sure the replicates are actually replicates. Yeah, I guess for this step, also the deep tool. For this step, deep tools is the one that you will use for generating this type of plots. So you don't need you don't need to know any programming language. So these are all command line that you can run and like even copy paste it and generate such plots. Of course, higher score of the for the correlation is better. And also it's important to mention that we restrict the, because CUT&RUN has near zero background, as we mentioned, outside the peaks, computing correlation across the whole genome would mostly be like, because there's nothing there, right? Like it's comparing 0 to 0, so which gives you low score for the correlation. So instead, we can restrict to peak regions. So you call the peaks. So instead of, like, that's why I brought this one here in this step, because you call your peaks and you're happy with your result, then you can say, okay, how similar my data are in terms of the peaks, because if you go with the BAM file, the genome-wide, it's gonna give you 0. But the peak files, they were actual signal in CUT&RUN. So you go with like you restrict to peak regions instead of the BAM files. So this is also important. And also we have IDR. That's another step for metrics like reproducible discovery rate, which filters your peak calls to only those consistently detects across replicates. So this is about the replicates. So this is like very similar to others, but this is important for reproducibility of the replicates. And once you're happy with all the metrics we talk about, you have to visualize your like you want to see, like from up to here you were seeing some kind of numbers, but now you want to visualize the signals, like how they are enriched in what region of interest that you're interested to see. So big file is the format that you need for genome browser tools like IGV. Like IGV, it allows you to upload big files, but UCSC, I think it's like a headache to make it, to visualize it in the genome track. So the BAM will convert to big so that you have a compressed files that you can visualize the enrichment of the peaks that you have, and you want to compare it to other regions. So you just search for the gene of interest and you check your data. But this step is very important because you have to visualize manually your data as well. Sometimes numbers are very different than the one that you are actually visualizing. So you have to drag both your big file and bed file on the genome browser like IGV, make sure the reference genome is correct, and go into like regions that's, I mean, they're like regions that are highly enriched in everywhere. So you can just test and see all of those regions have the actual peaks because those regions should be enriched anyway. So this is like a powerful sanity check before you commit to downstream analysis. As I mentioned, IGV, it gives you a very like user-friendly platform that you can visualize your data. And you can see how the peaks and the signals are actually aligned. So once you have your peaks, you want to see what motifs are present in those peaks. So your list of genome, like, peaks are the like genomic coordinates, right? So what you want to see, what DNA sequence does your protein prefer to sit on? And this is where the motif enrichment comes in. You use Homer. Homer is like an old tool that is very common. It doesn't tells you the footprinting. It just tells you what motifs are there, and they do like bunch of statistical analysis to tell you based on the data that you have. This motif is likely significant and is bound to this region. But again, this is based on so many statistical tests under the hood. So it's important to verify everything by yourself on the IGV. But this is like tool that gives you like a initial results. For example, you want to test like SOX2, for example, you expect to see SOX2 motif to enrich in the peaks. So that's kind of like quality check. There are like tools for TF footprinting, which are getting popular now, but they're like used for ATAG, like Tobias and all this Chrome VPNet and stuff. But for CUT&RUN, I think up to here is enough. And once you have your data and confirm the quality is good, sometimes you want to find out the differential analysis. So you want to apply a differential analysis on your data. So you have peaks and read counts. So you ask which regions are significantly more or less bound in one condition and it's not in another condition. So define handles that. Initially, it was built for ChIP-seq, but they use it for ATAG and ChIP-seq, CUT&RUN as well. So it merges all the peaks across all the sample into a common set of regions, count the reads in each, and runs a statistical test to find the differences. So it's probably incorporate your control, which is IGV in CUT&RUN, into the analysis, which is important to get to know what negative controls are. And they within the DiffBind, you have two different methods for statistical engine, which is DSeq2 or HR. You might heard of them, and they're used for count data type of thing, like RNA-seq. Also, they use the DSeq. Like DSeq is very conservative, whether false positive control. It's the default one, but HR is also sensitive, but it's like many more false positive as I experienced that. But you can test both of them and see what peaks are, shared between both of the methods, and maybe they are the ones that you're looking for. Sometimes the result is so good that both of the methods capture those differential accessible region or binding sites. So as I mentioned, not a single tool is perfect, so you have to try everything to make sure it actually answers your question. So I talked about all those steps. If you want to have them all into one single pipeline, it's this is the one, NF4 cut and run. Very easy to work with. You just make like your spreadsheets like other methods, and um, you know, run all at once. And it gives you every single QC metrics, peaks, and everything that we talked about. And you can change them, all the parameters as well. So it's very flexible, and I suggest you to not go through everything one by one. You just run this all at once, and once you get into a trouble, then you go back and see what was the parameters, like all the QC that we talked about, what are the what might cause the problem. There are like other websites that explain about cut and run data analysis, which I thought they are useful. And yeah, I guess this one is like annual workshop. I use their resources a lot. So every year in September, they have a workshop and they update their protocols. So it's kind of like a good tutorial for all the chromatin level data analysis. And yeah, that's it. And yeah, I put the first slide here if you want to scan the QR code for the first session. That's Waji perfectly covered everything in the wet lab. Yeah.
Thank you, Amin. Very
Yeah.
nicely done, and it was very,
Nice.
very interesting. I know for
Nice,
sure
nice.
I'll use some of those resources as well.
Yeah. It's so sometimes for cut and run you don't find that many resources because everyone thinks it's like kind of same as chip6, so they don't bother to uh, create another one. But sometimes it's important to know the differences between the protocols and apply those in your analysis. Yeah.
Absolutely. Thank you so much for sharing.
Awesome. No worries.
And the floor is open still for Q&As. Yeah, I guess we have a few in the Q&A chat box.
Mhm. So that's in the chat. Presentation. Can we share the presentation, Raji? I mean, I don't know.
Well, the whole video is going to be uploaded as well, so they're going to have
Mhm,
both the kind of like the slides animated and your narrative. So they're going to have
mhm,
both at the
mhm.
same time.
So yeah, they will be available soon on YouTube. So yeah, you will have everything there. Yeah. I feel like there were like some questions that was asked before, right? Maybe we can go through them.
Yeah.
Um, yeah.
I mean, I also
Someone,
have a question. Maybe someone
yeah.
who is like not a bioinformatician.
Mhm,
If someone who's interested in starting to analyze their own data,
mhm.
what would you recommend? What would be the first step?
The first step is to like sometimes I see colleagues that they think they should learn programming in general and then work with like all the tools and stuff that we talked about. If the focus is just analyzing your own data, what I suggest is just to learn the commands and what exactly they are, because all of them they have like good documentation, right? Um, Once you understand every step of your analysis, then you don't have to learn everything. You just have to ask what I need, for example, adapter training, and just search for that and run it on your data. That's what I think. But if you learn programming in general, of course, it's gonna be beneficial. But I see like some biologists, they're not even interested, and they struggle to they it's like in their mind always that they want to analyze their data, but they I don't know, they kind of hate or they don't like to deal with computer related stuff. So I suggest just go stick to one tutorial and that's it. Like don't switch between different tutorials here and there if you want to learn just like in general for future. But if you have your data, just ask the question and search for that. Now it's just ChatGPT, but even with Google, it helps you and go from there. Learn each parameter and what it does and all those things. And they're like BioStar forum, so you can ask questions there from real humans. Yeah, not like AI, but yeah. But sticking to one tutorial is important. If you open tabs so many, then you won't, I mean, based on my experience, you won't go like start it in any of those. Yeah.
Nice. Thank you.
Yep, yep, yep. No worries, no worries.
So it is 1 p.m. Central
Mhm.
Time. Um, if you have one question that you think would help most attendees, maybe answer that, and then we'll just wrap up so that
Mhm.
everyone's on schedule.
Yeah, yeah. Just one thing that I can mention that is if there is something like this NF core or encode pipelines, just stick to those and do not do all these steps by yourself, because then you'll have to deal with a bunch of random data that might not be, ideal for some people. I guess, yeah, it's, sometimes you generate so many results and then you get overwhelmed. So it's better to just, do it in a systematic way where all these tools are built for this type of issues like NF core, as I mentioned. It just, in every folder you have your result and that's it. You don't have to deal with renaming and everything, right? And I think I got, like, I don't know, my Q&A was not showing, but now I got another, like, 3 questions. How reproducible is CUT&RUN for non-histone protein? CUT&RUN is like, is actually like reproducible for like non-histone protein. Like you're talking about like transcription factors, even at like low input. So the key factors are, I think, antibody, as I heard, antibody quality, and a good antibody gives you like a clean, reproducible signal. So the main challenge with the transcript factor is that replicate can be real biology rather than just technical noise, right? So technically, the method itself is highly reproducible based on the experience, but the biology is that like the one that is different, then we all deal with. So I think having a best like antibody first is most important. Another question is the alignment rate. If the reference genome is not as good as human or mouse, could the successful experiment be less than 80%? It's a good question actually. Yeah. Based on that, exactly. That's 80% is more like assume well assembled references, like well assembled reference genome, like human or mouse. For organisms with like fragmented or like incomplete reference assemblies, lower alignment rates are expected for sure and acceptable even from perfect experiments. Honestly, I never like looked into that before, like in papers that they use different organisms, but I should go and check what are their percentage, like the rates of the duplication for, sorry, the alignment rates, sorry, for those organisms. But that was a very good question. So what is the difference between the tools setting for peak calling when using transcription factor CUT&RUN versus chromatin remodelers? Which one is more adequate in each cases? So the key difference is the expected fragment size and the peak width. So for transcription factors, TF bind at very specific short motif, right? So you expect narrow and sharp peaks, but for short fragments, typically filter for fragments under 120 base pair is ideal. Because there are some studies that are saying like how, like I talked about like different peak colors, but sometimes filtering based on fragments under 120, there was that study, like there was presentation actually. So the presenter, she said like she tested max2 with fragmented, like filtered for the fragments under 120, and the result was very different. Sometimes, you know, it's more like about, you know, the filtering that you're applying to the result. So the practical rule is if the protein acts as a single precise location, setting for narrow peak is ideal. And if it acts over a domain or a region, like a broad peak setting, that's, you know, you see in all the peak calling tools. Usually you have those settings in the peak callers, like broad or narrow. So based on that, you can just, you know, switch and see which one works. But yeah, some transcription factor like repressors, they're usually like broad. So we should also know that as well. Another question, is there any recommendation or tools for reduced batch effects? Sometimes it's not possible to do that at a time. There are like few approaches. Batch effects, yeah, it's always it's always a problem. So for the experimental level, so like always like have like same reference sample and control like across batches, like same across batches. So that's the first one. And spiking normalization helps a lot for since they like external reference independence of like it's like, you know, extended like independent, right? There are tools called, what is it called? It was combat, I guess, called combat. It's in R. So they use different methods to remove the batches, to like correct the batches. It works well for like batches that are known, and also like DSeq has its own built-in tool like design metrics. I think it's called design metrics. You can include batch as a covariant, yeah, as a covariant. So it's just, you know, directly applied to your analysis. It just, you know, it knows there are batches, so it goes for like correction and things like that. So yeah, I think if you look for the different tools, then they can help you for correcting the batches. I mean, it's everywhere, right? So it makes sense. I mean, yeah, but that was a good question. And the last one, are we out of time?
Yeah, we're a bit over time, but thank you so much. Your
Awesome.
answers were very thorough and very. You tried to complete as much
No
as
worries.
as possible.
Yeah,
Thank you
yeah.
again, and thanks everyone
Thank you.
for attending. Again, the presentation is going to be available online within a few days, and we hope to see you next time in our trainee-led series. Thank you.
Yeah. Thank you,
Have a good
everyone.
day.
Have a good one. Bye.